Thank you for joining us for Session 3, Using Mechanistic Modeling Approaches to Support Bioequivalence Assessments for Oral Products. The first talk of this session will be delivered by Dr. Fang, PhD, who is a senior pharmacologist and scientific lead at the Division of Quantitative Methods and Modeling, the Office of Research and Standards, the Office of Generic Drugs at the FDA. She has been with the FDA for more than 9 years. Dr. Wu is responsible for using modeling and simulation tools for reviewing pre-NDA meeting packages, ANDA consults, and control correspondences. Rebekah, Jereb, PhD, is a scientist at the Clinical Development Division at Sandoz. She will present next. Dr. Jereb has expertise in physiologically based pharmacokinetic modeling, IVIVC, IVIVR, and has developed various PBPK models for regulatory purposes, such as setting drug product specification criteria. She has published several research articles with focus on using PBPK modeling in generic drug development. Hannah Batchelor, PhD, will deliver the third talk of this session. Dr. Batchelor is a professor at the Strathclyde Institute of Pharmacy and Biomedical Sciences at the University of Strathclyde. She works on the design and manipulation of medicines to create age-appropriate drug formulations to maximize clinical efficacy in pediatric patients. Sumon Chakraborty is a scientific leader at the BioWaiver and Biocorrelation team in Apotex. He will deliver the last talk of this session. Mr. Chakraborty has more than 11 years of experience in the field of biopharmaceutics in generic industry. He has expertise in developing clinically relevant dissolution methods and bioequivalent safe space to address the specific questions pertinent to product development processes. The moderators of this exciting session are Tycho Heimbach, PhD, Biopharmaceutics Expert Director of Biopharmaceutics and Specialty Dosage Group at Merck, and Ethan Steer, PhD, who is serving as the Associate Director of Life Cycle Management at the Office of Clinical Pharmacology, Office of Translational Sciences at CDER FDA. Dr. Heimbach contributed to a priori PBPK modeling of more than 25 clinical trials and has authored or co-authored more than 15 INDs. He has authored and co-authored more than 60 peer-reviewed publications, 3 book chapters, and 2 patents. Dr. Steer is an expert in application of a variety of bioequivalence approaches to bridge formulation in generic and new drug applications. He is a recognized leader in guidance and policy development, including PSG, FDA guidance, and ICH guidance. Enjoy session 3.
Good afternoon, everyone. The topic of my presentation today is using PBPK model to support risk assessment for oral products from a regulatory perspective. This is my disclaimer. This presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. In this presentation, I will first introduce the regulatory questions that PBPK absorption modeling can help answer in generic drug development. Then I will present 2 research highlights, including using PBPK modeling to evaluate food impact on bioequivalence and using PBPK modeling to evaluate the impact of gastric pH on BE. As you may have known, the regulatory questions that PBPK absorption model can help answer in generic drug development include topics shown in this slide. They are all related to BE assessment, including evaluating the impact of changes in critical quality attributes, setting dissolution safe space, assessing the risk of formulation mechanism changes, etc. The PBPK absorption modeling can also be used to predict the impact of food and gastric pH change on BE, predict GI local concentrations, support waiver of in vivo studies, and BE assessment in specific populations. In this presentation, I will first introduce the research highlights on using PBPK modeling to evaluate the food impact on BE. This figure shows the effect of food on gastrointestinal physiology. Food can increase liver and portal vein blood flow, increased release of bile salt from gallbladder, increase gastric pH, delay gastric emptying time, and increase GI tract mobility, and increase volume and viscosity of the stomach and intestinal fluid, etc. In a recent collaborative research with the University of Washington, we investigated whether there is a trend in food effect based on BCS classification of drugs. We investigated 170 drugs with clinical food effect studies from the literature and the new drugs approved from year 2013 to 2019. The project found that drugs with significant increased exposure food effect were BCS class 2 or BCS class 4 drugs, while drugs with significantly decreased exposure food effect were BCS class 1 or 3 drugs. While assigning confidence based on BCS classification may be oversimplified, here I would like to provide some recapture points discussed in 2021 FDA CRCG workshop related to risk assessment of food impact. From the perspective of IQ consortium, high confidence of PBPK prediction of food effect are associated with BCS class 1 and 3 compounds, where a significant contribution of transport-mediated food effect can be ruled out. High confidences are also associated with a subset of BCS class 2 or 4 compounds, where the driving mechanism of food effect can be attributable to changes in solubility in the fat state related to changes in GI physiology. While the main driver for low confidence in predictions associated with salt form, effect on microenvironmental pH, changes in hydrodynamics due to viscosity in presence of food, and in vivo solubility change, for example, for compound that undergo precipitation. Under such situations, a middle-out approach can be utilized with high confidence using a clinical anchor study. From generic drug side, we conducted an internal research and used the oral PBPK model of acyclovir immediate release tablet for assessing the impact of food intake and expiant on the BE of generic acyclovir IR tablet using virtual healthy subjects and virtual bioequivalence trials. Our developed acyclovir PBPK model was validated with PK data from literatures and approved ANDAs. As shown in the right figure, acyclovir oral PBPK models successfully simulated the impact of food for 800 mg IR tablet with a delayed Cmax. Virtual BE was conducted for acyclovir IR tablet under both fasted and fat condition, indicating food appears not to impact the bioequivalence results for this case. So based on the discussions in the 2021 FDA CRCG mechanistic modeling workshop and research experiences, here summarize the considerations when using PBPK to evaluate food impact. For example, the role of pH and bile salts on dissolution rate and extent of drug release need to be taken into consideration while deciding whether a biorelevant dissolution media would be advantageous over QC dissolution media. Further research is needed to assess the probable role of dissolution apparatus and mechanical setup, for example, apparatus type, pedal speed, used in generating biorelevant dissolution data to mimic fat condition. In addition, research related to the impact of food on transporters is needed in order to leverage the full potential of PBPK model in food effect prediction. Default between subjective variabilities, BSV, may be modified if proper justification and the data related to the modified variability are available. For highly variable drugs, BSV may need to be incorporated according to the clinical data. Further, when food effects of orally administered drugs are mediated by the gastric emptying, GIPH, PBPK models may predict the impact of food intake more accurately compared to more complex mechanisms, such as transporter, metabolism, food-drug complex formulation, and formulation-mediated food effect. So relevant research is needed on understanding the above mechanisms, including formulation-mediated food effect. Also, further research is needed to understand and develop mechanistic relationship between types of meal and their impact on oral absorption. This table summarizes the highlighted cases where PBPK absorption modeling had impacts on regulatory decision-making, including a case example of using PBPK absorption modeling and simulation to evaluate the impact of food on BE based on in vivo fasted and pilot fat BE study, which I will introduce more in the next slide. Here is the case example about using PBPK absorption modeling to evaluate food impact on BE for drug X oral tablets including active pharmaceutical ingredients with amorphous solid dispersion form. A mechanistic absorption model was developed based on literature data and results from pilot BE studies and used for predicting the bioequivalence and the fat conditions. We evaluated the risk and complexity of the formulation of the proposed product and identified the major concerns, limitations of the developed PBPK model. For example, there is a lack of supporting information related to formulation design, manufacturing process, API characteristics, expense, and quality attributes of the drug product that may significantly impact the in vivo dissolution and the bioavailability of drug. Also, the model validation step is based on BE studies, which demonstrate BE among the batches tested, challenging the model with in vitro and in vivo data, which show the lack of BE and or batches with different release rate to support robustness of the established PBPK model is recommended. In summary, research projects and the regulatory submission use PBPK modeling to assess risk of bio-inequivalence attributable to food intake and or provide justification of not conducting fat BE study. To fill in existing knowledge gap and gain more experiences, relevant grant and contract are funded by GUDUFA. Here listed two recently funded grant and contract projects. The grant project development and validation of a best practices framework for PBPK analysis for biopharmaceutical applications in support of model-informed biowaivers of fat state BE studies for BCS Class 2 drugs was funded to University of Florida, and the contract project, disintegration and dissolution of solid dosage forms and influence of food-induced viscosity on its kinetics, tools, and methodologies for bioequivalence as substitutability evaluation was funded to join Gutenberg University. Both projects are supporting the evaluation of food impact on BE. Another topic I would like to introduce today is using PBPK modeling to evaluate the impact of gastric pH change on PK and the potential impact on BE. For generic drugs, additional BE studies in subjects with altered gastric pH may be needed when there are formulation-dependent gastric pH-mediated drug-drug interaction. A recent published product-specific guidance for palbociclib oral tablets recommends 3 in vivo BE studies, including a fasting BE study in the presence of acid-reducing agent. From new drug side, a guidance for the evaluation of gastric pH-dependent drug interactions with acid-reducing agents was issued in 2020. The figure on the right showed a framework to assess clinical DDI risk with ARAs for immediate-release products of weak-base drugs, including using dose and solubility as well as dissolution comparison at different media conditions. In conjugation with the assessment framework outlined in this figure, PBPK simulations can sometimes be used to further assess the potential for pH-dependent DDIs. To investigate how solubility and the dissolution profile comparisons and the different pH conditions can be used to predict gastric pH-mediated drug interaction potential, we conducted research. We analyzed 67 new molecular entities that are proved from the year of 2003 to 2019 with solubility information and the different pH and dissolution profiles generated in pH 1.2, 4.5, and 6.8 aqueous media. Similarity factors were used to compare dissolution profiles at different pH for pH-mediated DDI prediction. We use F2 less than 50 predicts positive DDIs. Prediction accuracy was calculated based on the outcome comparison between predicted and observed DDIs. As shown in the table, to prediction accuracies using dissolution profiles under both fasted and fat conditions were comparable to the prediction using solubility and clinical dose, with 72.5% prediction accuracy versus 80% accuracy under fasted condition. And they have similar prediction accuracy under fat condition with 66.7%. Then, in another research project, we investigated whether PBPK modeling can be used to evaluate the potential of such pH-dependent DDIs for 4 weight-based drugs, including tapentadol, darunavir, erlotinib, and saxagliptin. PBPK models were developed and optimized using PK data following oral administration without ARAs, which were then verified with data from additional PK studies in the presence and absence of food. The models were subsequently used to predict the extent of DDIs with ARAs co-administration by changing the gastric pH to 6. This figure shows representative model prediction results. We can see, using both GastroPlus and Simpson PBPK models, the simulated plasma concentrations can capture the observed data well after administering single oral dose of 80 mg tapentadol, 100 mg saxagliptin, 600 mg darunavir with 100 mg ritonavir under the fat condition, and 150 mg erlotinib, respectively. The validated PBPK models were then used to predict drug exposure and pH-dependent DDI following a single-dose administration with and without concomitant omeprazole. This table shows that the model predicted the ratio of Cmax or AUC in the presence and absence of ARAs matches well of the observed values for tapentadol, saxagliptin, and darunavir. While for erlotinib, it appears a bit overpredicted the ratio of Cmax and AUC in the presence and absence of ARAs. In summary, the results suggested that PBPK models developed could adequately describe the lack of the effect of ARA on the PK of tapentadol, darunavir, and saxagliptin, and could qualitatively predict the effect of ARA in reducing the absorption of erlotinib. Using solubility, dissolution, and modeling approaches can help evaluate the impact of gastric pH on drug exposure or bioavailability. For generic drugs, when there are formulation-dependent gastric pH-mediated DDI, additional BE studies in subjects with altered gastric pH may be needed, especially under certain high-risk situations. For example, high risk exists when test products and the compared products contain different levels of pH stabilizing or modifying excipients. To demonstrate that BE study in a gastric pH altered situation may not be needed, scientific justifications, including pH solubility profile, comparative dissolution testing at multiple pH, and PBPK modeling may be used. In conclusion, PBPK modeling has been used to assess the risk of bioinequivalence attributable to food intake, solubility dissolution characteristics, and PBPK modeling can be used to predict the impact of gastric pH on drug exposure and may be used to predict the impact of gastric pH on BE. Further investigations is warranted to demonstrate that PBPK modeling can be used to assess whether a BE study in a gastric pH altered situation is needed. As demonstrated above, we have conducted research related to oral PBPK modeling through both internal and external projects. Here listed a couple of recent publications based on these research projects. You may refer to these publications for details. With that, I would like to thank all the people listed here for their support and hard work, especially the reviewers, scientific assessors involved in the regulatory work and research. Also, special thanks to those authors who are involved in the publications that I mentioned in the slides. This concludes my presentation, and I will be back later to answer questions during the panel discussion. Thank you for your attention.
Welcome, everyone. The purpose of this workshop is to discuss opportunities and best practices for incorporating modeling and simulation approaches into generic drug development programs and regulatory submissions. My talk will focus on PBPK modeling used during drug product development to support risk assessments for oral drug products, and also possibilities of using PBPK modeling for regulatory purposes, such as waiving FAT-BE studies. The opinions expressed herein are solely mine and do not represent statements or opinions of League Pharmaceuticals, Sandoz Pharmaceuticals, or Novartis. First, I'll focus on PBPK modeling used during drug product development to support risk assessments and present a case study. Then I will continue with the current state of PBPK modeling being used for food effect predictions and what options there are for waiving FAT-BE studies using PBPK modeling. In the end, I will also present a case study on this last topic. To guide PBPK modeling activities, we usually follow health authority guidance listed here. 2 US FDA guidance, 1 EMA, and lastly published Japan guideline on PBPK. First US guideline, EMA, and Japan guideline recommend how to prepare a report of a PBPK analysis to be submitted for regulatory purposes, and what details on model development and validation to include. Most relevant for the continuation of this presentation is second FDA guideline, the use of PBPK analysis for biopharmaceutics application, as it focuses on drug product quality attributes and a mechanistic understanding of their interaction with physiology to affect in vivo drug performance of oral products. For this analysis, also termed PBBM, physiologically based biopharmaceutics modeling, is being used. Physiologically based biopharmaceutics modeling can be a helpful tool to assess potential bioequivalence risks and predict the outcome of bioequivalence studies if properly developed and validated. The use of PBBM in generic drug development is widespread, ranging from API or formulation property selection, assessment of effect of dissolution changes on drug in vivo behavior, setting of clinically relevant specifications, in vitro in vivo correlations, food effect predictions, and assessment of DDI effect of acid-reducing agents. The first case I'm going to present is a PBPK model for a BCS class 3 drug with high solubility and low permeability. During development, we discovered that test and reference products do not have similar dissolution at low pH. However, the products were bioequivalent in vivo. We therefore used PBPK model to assess the biorelevancy of in vitro dissolution at pH 1.2 and the impact of non-similar dissolution profiles on tablet in vivo performance. We developed a PBPK model in GastroPlus based on available literature data, some experimental data, and internal bioequivalence study results, and predicted plasma concentration profiles and PK parameters for test and reference products. The model was developed gradually, using available intravenous data for fitting distribution parameters. Some optimization was done during model development, such as lower volume of fluid in small intestine and colon according to the values reported in literature. For the simulation of tablet test and reference products, we used in vitro dissolution at pH 1.2, 4.5, and 6.8. To validate the model, some parameter sensitivity analysis was performed on API particle size, solubility, permeability, first pass extract, etc. Also, virtual population was simulated, and the results of virtual trial were compared to data observed in vivo. After the model was validated, it was used to simulate in vivo dissolution using different stomach transit times. As mentioned in previous slide, we used dissolution at 3 pH values, 1.2, 4.5, and 6.8 for the simulations. Z-factor was fitted to these dissolution profiles, and Z-factor versus pH table was used for the simulations. From the figure, we can observe that dissolution at low pH is slow and dissolution at higher pH is relatively fast. Here are the results for model validation. The simulated plasma concentration profiles after intravenous administration of 2 different doses capture the in vivo observed data well. Also, prediction errors for Cmax and AUC are all below 10%, which is a good result. Here we can see that single simulation for the test tablet formulation, which capture the observed data well. Also, virtual trials capture the variability observed in vivo with minimum and maximum Cmax and AUC simulated values comparable to the in vivo observed values. Thus, we considered our model validated. Then, we simulated a hypothetical in vivo dissolution if the tablet would stay in the stomach for infinite time, that is, at low pH without absorption. The simulated profiles show very slow and not complete dissolution and difference between test and reference. This is not in line with in vivo BE study results where products were bioequivalent. The dissolution at pH 1.2 does not seem to reflect in vivo behavior and is thus not considered biorelevant. On the other hand, simulation of in vivo dissolution with tablets transiting through gastrointestinal tract and absorption happening shows similar dissolution of test and reference formulations. This is in line with in vivo bioequivalence results. Now, coming to the second part of my presentation, focusing on modeling of food effect. According to EMA guideline on the investigation of bioequivalence, the study should be conducted under fasting conditions, as this is considered to be the most sensitive condition to detect a potential difference between formulations. Studies in both fasting and fat conditions are required only for products with specific formulations characteristics, such as microemulsions or solid dispersions. In contrast, US FDA recommends that applicants conduct fat bioequivalence studies in addition to a fasting bioequivalence study for an orally administered immediate release product. The exception of only doing fasting study can be made when the reference labeling states that the product should be taken on an empty stomach or when serious adverse events are anticipated with administration of the drug product under fat conditions. The question that rises after reviewing all available information on food effect prediction with PBPK modeling is, can PBPK modeling be used to weigh fat bioequivalence study? Food has various effects in the gastrointestinal tract, from changing the environment pH, volume of fluid, concentration of biosalts, and transit times of formulations. In addition, there is also a possibility of binding of drug substance to food components, degradation of drug substance, and impact on transporters and enzymes. All these effects result in positive, negative, or no impact on drug bioavailability and pharmacokinetic parameters. In general, we could expect an impact of delayed gastric emptying on Cmax of a BCS class 1 drug. Also, for low-soluble molecules, we could expect increased solubility due to the effect of biol salts and changes in volume of fluid. PBPK models are more or less capable of predicting food effect. There have been many publications in the recent years reporting the evaluation of how well PBPK models are able to predict food effect. Kesisoglu proposed a workflow to streamline food effect assessment for different BCS classes. He emphasized that in vitro methods should be relevant to specific food effect. He also reported that model optimization is a usual part of model development. Cheng and Wang emphasized that when predicting fat state, the model has to account for physiological changes in the gastrointestinal tract as well as changes in drug properties, for example, using biorelevant solubility in fat state media. The models have some constraints, such as inability to capture lymphatic uptake or more complicated mechanisms of negative food effect, such as degradation, inhibition of transporters, increased viscosity, etc. Riedmeier et al. Performed analysis of de novo PBPK model development and food effect predictions for 30 compounds. They evaluated the confidence in predicting food effect. High confidence was achieved when food effects included increased solubility due to effects of bile acids and changes in gastrointestinal transit times. Moderate prediction confidence included food effect caused by lymphatic uptake or precipitation. Low confidence was related to changes in viscosity and microenvironment pH. Pepin et al. Explained in more details how a combined bottom-up and top-down, that is middle-out approach, was used to increase confidence in some of these food effect predictions. Some adjustments of fluid volume had to be made, as well as permeability fitting on data from study in the fasting conditions. Wagner et al. Provided some recommendations for improving predictive performance of low confidence food effect models. They emphasized the importance of using biorelevant solubility in both fasting and fat conditions to account for changes in pH with time, which is especially important for compounds with pH-dependent solubility. Sometimes also permeability changes from fasting to fat conditions are important. Some in vitro methods to evaluate precipitation were also suggested. Riedmeier summarized all the work done in previous publications. She suggested that following validation with fasted data, models can be successfully applied to predict the fat state in lieu of a dedicated clinical food effect study, especially for BCS1 and 3 compounds. There still remain some gaps, for instance, modeling of changes in transporter on enzyme kinetics in presence of food, or biopredictive in vitro methods to assess negative food effect. Anyhow, a middle-out approach can be applied with high confidence where the fat model is verified with a clinical study and subsequently extrapolated to additional food effect questions, for example, following formulation or dosage changes. This sounds like an option for generics to pursue. During generic drug development, we usually have a lot of clinical data available to develop and validate PBPK models. We could apply middle-out approach, as usually food effect studies are performed by the originator. The most important questions generics have to deal with is how to incorporate the difference between formulations, that is, between test and reference products, into the model. We conducted an analysis on 6 case studies to evaluate the approach if the results of bioequivalence study in fat conditions can be predicted based on PBPK model developed on data from bioequivalence study in fasting conditions and known food effect. The difference between test and reference was entered into the model using different dissolution profiles or difference in API particle size for test and reference or different solubility values. Disposition parameters for these 6 cases were determined using intravenous data when available and in one case using oral suspension data. Some optimization was applied, mainly to reduce volume of fluid in gastrointestinal tract, some changes in gastric pH, precipitation time, and absorption scaling factors for BCS class 3 drug. Permeability was entered based on measurements in KCO2 cells or MDCK. Then virtual bioequivalence studies were ran. Number of subjects was the same as used in vivo bioequivalence studies. We simulated 10 studies per fasting and fed condition. The variability in physiological parameters was the same as observed in vivo. Model validation was performed based on all available clinical data, which could result in larger % prediction error. In addition, some proper scientific justifications were provided for the selector parameter values or any optimization performed. Some prerequisites that we identified as needed for successful prediction of bioequivalence in fed state were BCS class 1 or 2 drug with known food effect, preferably linear pharmacokinetics, reliable estimation of disposition parameters, preferably intravenous data if available, available bioequivalence study data in the fasting conditions, and estimates of PK parameters variability are also needed. When all these are checked, the confidence in the prediction is high. The second case I'm going to present is a fed bioequivalence study prediction for a BCS class 2 drug. We developed the model based on published clinical data, intravenous, capsule, and tablet, including different doses. We also performed fasting and fed pilot studies with 2 different test batches and reference batch. Results of pivotal fasting B study were also available. To use biorelevant dissolution method for prediction, 2 different methods were used. For fasting simulations, simulated gastric fluid with pH 1.2 was used to mimic acidic conditions in the stomach. For fed simulations, buffer with pH 4.5 and SLS was used to account for pH in the fed stomach and effect of bile salts. The results of 10 simulated fed bioequivalence studies are presented in the figure. All 19% confidence intervals for Cmax and AUC are between 80 and 125% and predict bioequivalence of test and reference formulations. This was in line with the in vivo BE study conducted. There were some model deficiencies identified by health authority to whom the model was submitted, and some suggestions for improvement are presented. The model was not considered to be properly validated. We should have different batches with different release rates, preferably a batch with non-bioequivalent result in vivo. Also, in-house experimental determination of solubility in biorelevant media should be performed. Lastly, the dissolution methods were not considered to be biorelevant, and 2-stage dissolution with simulated gastric fluid and FASIV or FASIV were suggested. To conclude, PBPK modeling is useful during generic drug development. It has been widely used for internal decision making. However, for regulatory applications, there are high standards for model development and validation. These are sometimes difficult to achieve as there may not be enough data available for model development and validation. Discussion between industry and regulatory is crucial to make regulatory application of PBPK modeling a reality. In the end, I would like to thank all my colleagues at Sandoz, both in SDC Slovenia and globally, for many scientific discussions about modeling. Here are the references used during the presentation. Thank you for listening. Please provide any questions in the chat. I will also join the panel discussion.
Hi, my presentation here is going to talk about the use of oral physiologically based pharmacokinetic modelling to support bioequivalence evaluation for paediatric drugs. My name is Hannah Batchelor. I'm a Professor of Pharmaceutics and Biopharmaceutics in Strathclyde, which is Glasgow, Scotland, UK. The basis of my talk comes from a meeting that was held 5 years ago at the AAPS in San Diego, where we were exploring whether bioequivalence established in an adult population is relevant for paediatrics. On that day, we really queried these questions about whether there was a way to predict at-risk paediatric products. What we were seeking was tools, in vitro and in silico tools, to de-risk the extrapolation of bioequivalence in adults when projected into paediatric populations. A project was set up jointly between myself as an academic and the FDA with the key goals of identifying generic pharmaceutical products most at risk of suboptimal therapy in paediatric patients. And to do this, we used literature to scope the evidence and then use some in vitro and in silico models to really understand the risks. And it's that final part I'm going to focus on today. The objectives of the project were twofold. There was a first part to really explore what data was out there. One of the issues in understanding the risks of suboptimal performance in paediatric products is the limited clinical data that is publicly available. So the first part was to generate a database of all that data that we could then mine. And the second part I'm going to go into in more detail, which is the dissolution and PBPK modelling. So this first part was development of this database, and I'm going to just briefly skim over this to allow time for discussion. The database was generated by a literature search using a range of keywords over a range of different databases that are publicly accessible. We applied some inclusion and exclusion criteria and ended up with 78 clinical studies that contained data from paediatric populations. Of these studies, 40 showed bioinequivalence, and for the purposes of this talk today, that means that two products were not equivalent in terms of their exposure, either due to the population changes or the product changes. So this here shows from those 40 studies the reasons that the authors gave for that bioinequivalence that was observed. So sometimes it was related to age-related absorption, sometimes it was the study design and the sample size. Pediatric studies can't be conducted in healthy pediatric populations, so we have to use patients with the disease, which can limit the sample size and also affect the variability observed in a clinical data. Sometimes the effects were due to the drug substance or formulation, so perhaps comparing a chewable pediatric formulation to a conventional adult tablet where you might expect a difference in the relative bioavailability. And here we're interested in is the ratio of difference observed in adults translatable to the ratio of difference observed in pediatrics, and how does that affect the extrapolation? And again, there were some age-related disease effects due to the need to conduct studies in non-healthy children. This slide just covers the same aspects in numerical form. And if you are interested in more detail, we have published this and the paper is available with a link just here. The second part, which will be the focus of this talk, was about the development of these in vitro and in silico methods. So we're using dissolution for in vitro and physiologically based pharmacokinetic modelling for in silico. We know that dissolution is a commonly used test during pharmaceutical industry and in regulatory guidance to evaluate the comparability of two formulations, either for bridging or for a generic to an innovator product. We also know that there's variability in gastrointestinal fluids in children compared to in adults based on their composition. And we also know that there's differences in the volume that can be observed in children compared to adults. There's publications on this that have come out of my group that I'd be delighted to share for anyone interested. So one of the things we wanted to explore was whether we could use dissolution testing that integrated these compositional and volume changes to create a biorelevant system that might better predict performance in pediatrics and be superior in terms of integration into the physiologically based pharmacokinetic modelling tests. At the moment, there are well characterised and well recognised gold standards in terms of the adult simulated gastric fluids and simulated intestinal fluids from the fasted state. The work that we've done and the references here on paediatric fluids gives us these criteria as limits to create our paediatric simulated intestinal and simulated gastric fluids. These are not as well recognised as the adult ones. Key on this slide is the bile concentrations, where the bile salt in the gastric and in the intestinal fluids is much lower than the equivalents observed in the adults. So bile salts are the surfactants used that can help solubilize the drug, so the compositional changes might well affect the dissolution in a paediatric population. In our studies, we also measured which bile salts were present, so we could replicate the relative ratio of each of those, because dependent upon the specificity of the product present will affect the colloidal structures formed, which subsequently affects the solubilization profile and the dissolution. Part 1 highlighted those products at risk of bioinequivalence and gave us 4 case studies to work with. And the data I'm going to present today is really looking at carbamazepine and how that is affected in the in vitro dissolution and the PBPK modelling. So we created a series of dissolution plans to understand which dissolution profile we should be integrating into our PBPK software. So adult fasted state simulated gastric fluid and fasted state simulated intestinal fluid are the most common fluids used using USP apparatus, so the standard pharmacopoeial apparatus. We looked at different volumes on those to reflect the difference in volume seen in paediatrics compared to adult populations. We also then compared these adult media to paediatric media to look at the compositional changes. Because there's currently no gold standard for these paediatric simulated gastric fluids and intestinal fluids, we wanted to compare a range. I'm just going to describe them briefly here. So the paediatric gastric and intestinal fluids that we used here contained 14 bile salts in the ratios that we had identified from the characterization of human fluids from paediatric populations. We also then wanted to compare the same fluids but at a much lower level of bile salt, so we diluted these in half to give us a 50% bile salt to give us a worst case scenario. We then also created the same simulated fluids, but rather than using the 14 bile salts, we used a single bile salt of taurocolic acid. And it's a single bile salt that's used in the adult simulated gastric and intestinal fluids. So, the use of our PBPK in conjunction with clinical data and dissolution was really to understand which dissolution profile best predicted any of the pharmacokinetic profiles we could get from the literature in both adult and paediatric populations for carbamazepine. We wanted to use this to then propose the most suitable dissolution methodology that could be integrated when looking at extrapolation into a paediatric population. So this is a data-heavy slide, and I'm going to just spend a moment talking through it. In this slide, we are looking at the effect of dissolution volume from 900 to 50 millilitres using the adult gold standard fasted state simulated gastric fluid on the 100 milligram carbamazepine tablet. We can see here at the highest dissolution volume, we're getting the most complete dissolution compared to the 50 millilitres here at the bottom. We use the F1 and F2 statistic to look for statistical similarities or differences between the profiles. And in this table, we can see that actually statistically, the dissolution at the 900 and the 500 was similar, whereas there was differences between most of the other volumes. And again, this is shown, the significant differences are shown by these lines here. So in summary, the media volume affects the dissolution for carbamazepine, which may be anticipated as it is a poorly soluble drug. This slide looks similar, but this time we are looking at the intestinal fluid rather than the gastric fluid, but we see the same pattern of data. As the volume decreases, we get less dissolution that might be expected, and it's actually the same pattern of similarity based on that F1, F2 statistic between the volumes. This slide here is showing us something similar but different. So we've just explored the effect of volume on dissolution, and this time we're looking at the effect of media composition. So we're comparing 4 different simulated gastric fluids, the adult, to the paediatric ones that I described previously. And all of these were compared in a 200 milliliter volume because we're really looking to replicate the paediatric intestine here, and that was a sensible volume based on the volumes observed in our other study. You can see here there's no difference on the effect of the composition of the fluid in gastric media for any of these fluids. This is the same study, but this time rather than using the gastric fluid at 200 millilitres, we're looking at the intestinal fluid. This time we did see a difference between the adult media, which contained the highest bile salt concentration, and the paediatric media that contained 50% or the lowest concentration of the bile salts here. So there was a limited effect of the composition affecting the dissolution in the simulated intestinal media, but only at the extreme cases. So these slides here are comparing now a reference to a test, so an innovator to a test carbamazepine tablet in the gastric and the intestinal media. Here we're trying to look at differences that might be predicted in adults. So when we're using 500 millilitres of media, are there differences? The F1, F2 did actually show some subtle differences between the reference and the test product in the simulated gastric media at 500 millilitres. The right-hand panel is showing the similar data, this time using 200 millilitres to look at a paediatric population. And again, difference was observed in the simulated gastric fluids. This didn't cause me too much concern, as we generally look at the intestinal fluids as being the bigger predictor of differences in absorption. So I'm going to move into the integration of that dissolution into the physiologically based pharmacokinetic software. We used SimSip software. These are the details for our studies, and we were going to integrate our dissolution data into the ADAM model of the SimSip simulator and look at the model prediction accuracy to really understand which dissolution inputs are giving us the best predictions. For reference, here are the model inputs, but I'm not going to go through them in detail in the interest of time. Happy to share these with anyone interested. So here is some of the data. The blue crosses are showing clinical data we got from the literature here in adults. And the simulated line, the green line that lies behind, we have integrated our dissolution data using the 500 milliliter volumes of our adult simulated gastric and simulated intestinal fluid. And what we can see here is actually the simulation is really nicely overlaying the clinical data in 2 different studies. This gave me some confidence and modelling was good. We then integrated various other data that we could get from the literature here. So these are all the studies that we integrated, and we compared our predicted data using the PK parameters of the area under the curve and the Cmax to the observed data. And what we've got in this final column, which I'm going to just highlight, is our percentage prediction error. And where we've got a value of less than 20%, or where these are green, we were happy with our modelling. So there's one case where it wasn't, but otherwise we were generally pretty happy that using the 500 millilitres of the simulated gastric or simulated fluid provides a really good fit to the clinical data. This slide we're doing the same thing, but this time we're overlaying some of our data with paediatric populations. So in this case we've got data from children that we've put onto this graph, and we are overlaying here our dissolution data using the 200 milliliter volume of the different paediatric media that we had. So again, we've tried to compare our predicted values to the observed values, and we got pretty good values for our percentage prediction error. There was one study where the Cmax wasn't perfect, but otherwise the dissolution data using the 200 millilitres of the paediatric media was mapping well to our clinical data. So this was suggesting it's a good input for this data. We went back and looked at that dissolution data that we generated for the different volumes of the adult media. I've said previously the paediatric media is still under development, whereas the adult simulated gastric and simulated intestinal fluids are well-known, well-regarded fluids that we can use easily in development. So this chart here is showing when we simulate using the adult 500 millilitres and overlay to the paediatric data, we're miles away. When we're using 200 millilitres, this rusty orange line, we're reasonably close. At 100 millilitres, we're too low, and 50 were way too low. The dashed blue line for reference is that paediatric fluid at 200 millilitres. So we can see the paediatric fluids are still the best predictors. If you're going to use adult volumes, then 200 millilitres is reasonable. This is 100, so perhaps somewhere nearer to 150 millilitres might be even better for the adult media. And again, we've just got the percentage prediction errors up there, and it does show that some of our media with the sodium taurocholate is better in this example compared to the adult. Again, this is a very data heavy slide, and I'm conscious of time when I'm explaining this. So here we ran virtual bioequivalence studies. So this allows you to input the dissolution data and run a virtual bioequivalence study that accounts for the variability in your population. So in our adult populations, when we integrated any of our 500 millilitres data to this, so either using the intestinal fluid alone or a combination of the gastric and the intestinal fluids, we were meeting our bioequivalence criteria no matter the number of subjects or the number of trials. For the paediatric subjects here, when we were integrating just our paediatric intestinal fluid, we were getting good data for our bioequivalence. But when we started to also incorporate the simulated gastric fluid, we were just marginally missing some of those bioequivalent targets. So the target is 80 to 125%. So the misses were marginal. We've got to accept that those are misses in the virtual bioequivalence. One other thing to note here is when we undertook these simulated clinical studies, we gave the adults a 240 milliliter volume of fluids with their tablets, as is normally recommended in the adult clinical protocol, whereas for paediatrics we gave them a smaller volume of fluids because they were younger. This also replicates what was given in clinical studies. So again, it just suggests that for a virtual bioequivalence simulation, I would recommend using a 200 milliliter volume of just the intestinal fluid to predict whether your generic and innovator product are likely to be the same in a paediatric population. In conclusion, we can show that carbamazepine dissolution is more sensitive to volume compared to the composition of your intestinal fluids. The integration of 500 millilitres of adult simulated fluids was giving a good match to clinical data, and 200 millilitres of our paediatric simulated intestinal fluids, again, likewise giving a good match. If you desperately need to use adult data, I would recommend a volume of 150 to 200 millilitres as being superior. And the virtual bioequivalence was showing good data, and it was really dependent on our dissolution inputs, which is perhaps unsurprising. My last 5 seconds, I definitely want to thank Gopal for his work on this project, as well as everybody else listed on this slide, for all of your input into the discussion of the project. I'll happily take questions in the panel, and thanks so much again for your attention.
Hello everyone, I am Sumon Chakraborty from Apotex, and today I'll be presenting the different approaches in establishing BE safe space for OSD. I would like to thank the CRCG and FDA for the kind invitations to present some of the case studies in this exciting session. Let's start with the disclaimer first. The views expressed in these presentations are my own and do not represent any views from Apotex. These are the agendas for today's discussions. The definitions of and the applications of BE safe space, different methods which are available to establish the safe space, and few case studies of utilizing the physiologically based biopharmaceutics modeling approach in generic industry for internal or regulatory decision making. A safe space is basically defined by the boundaries demarcated by in vitro specifications. It could be a resolution or any other drug product quality attributes for that matter, within which the drug product variants are anticipated to be bioequivalent to the pivotal biobatch. In the right side of the presentations, the resolution data and the plasma profile of some of the batches were presented, which includes the biobatch, the non-biobatch, and the reference formulations. Now, in absence of modeling and simulations approach, the resolution profile between the brand and the biobatch is the safe space, which is pretty much narrow, as in most of the time we try to manufacture the pivotal biobatch which is as close as possible to the brand formulations. Now, with the additions of a non-B batch, we could able to expand the limit of knowledge space. And also, since the method gives a rank order correlations, meaning that the slower dissolution rate produces the slower the plasma profile. So now with this rank order relationships, we can establish the PBBM, and we can expand the safe space beyond the biobatch data, and the safe space is typically between the biobatch and the non-biobatch. These are the different areas where we can apply the concept of the safe space for either for the internal or regulatory flexibility. This is the area where the safe space is utilized by the generic company frequently, and help us selecting the BE lot for pivotal study. There are a couple of more areas where the concept of the safe spaces are being utilized for the regulatory decision making. For example, the dissolution dissimilarity for the additional strengths for biowaiver, to provide the specifications, for example, the dissolutions or FESD, and in case of major post-approval changes. The generic companies are now utilizing the approach or the concept of PBBM to assess the impact of food effect, especially for immediate release drug product with BCS 1 and 2, and to assess the impact of pH mediated drug-drug interactions for immediate release product with BCS 2 week base. In this slide, we are going to talk about the different methods available to establish the BE safe space. The first one is very simple and a very traditional one. And this is usually very narrow because the impact of critical bioavailability attributes were not evaluated in vitro and in vivo, and hence very limited data available. And the safe space may be unnecessarily too tight because the relevance of the narrow space is unknown. In the second approach, we have the data from two test formulation variants where the critical bioavailability attributes were varied, considered and varied to manufacture those batches, and the safe space is decided based on the BE data of those two test formulations. Despite the fact that the two test formulations are having a different dissolution profile. The third approach is a conventional IVIVC, and in this approach we need the data of at least two test formulations uh, where the critical bioavailability attributes with the help of the dissolution data could be linked to the in vitro release profile by mathematical equations. In case of immediate release product, where it is usually very difficult to establish the level A IVIVC because of the significant difference in the time scale between in vitro and in vivo, and in that case a level C could be an useful alternative. However, if you want to use the level C IVIVC, then we need to have at least a 3 formulation data, because the relationship which is basically used for correlating in vitro and in vivo or linear regressions, and we need at least 3 different time points to calculate a straight line equations or regressions equations. In the last approach, the CBA dissolution data, physicochemical data and the physiologies are integrated to establish the BE safe space. In this last two approach, the safe space are basically expanded beyond the BE batch and help us developing a clinically relevant dissolution method and the specifications. In this slide, we are going to talk about the general workflow for the physiologically based biopharmaceutics modeling. So before we start developing the model, the objective of the model has to be decided first, and it could be anything from the list that discussed in the slide number 4. We will have to collect the data either from the experiment or from the literature, from the API specific formulations, physiology and disposition parameter, and then integrate all of this to develop the base model. The disposition model to be developed first, and then the absorption model using the dose linearity study from the literature as an observed data. Then the model development using the pivotal biobatch, and in this stage we are basically integrating the in vitro dissolution data and justify the dissolution model selections for the future use. If the model does not meet the criteria of validations, right, then the optimizations of certain important input parameters to be performed here, and optimizations to be supported by a parameter sensitivity analysis, right? And it has to be scientifically it should be scientifically rational and not just by the best fitting approach. And then in the next stage, we typically incorporate the data, the in vitro data of the second in-house batch, which is basically a non-bio batch from the pilot study, and check if the model is able to reject the non-bio batch based on the prediction error or based on the virtual BE trial. And then once the validation criteria are satisfied, we are allowed to use this model for its intended applications. Then typically we generate the virtual dissolution profile to expand the BE safe space, and the limit can be decided based on either of these two approaches. So in first approach, which is basically taken from IVIVC guidance US FDA 1997, in this approach the difference in predicted PK parameter between the target, where here the target is the pivotal biobatch, and the virtual profile should be no more than 20%. And in the second approach is basically from the PBPK guideline FDA 2020. In this approach, the 90% confidence interval of the point estimate between the virtual batch and the target profile should be within 0.8 to 1.25. In this slide, we will talk about the eligibility and limitations of different the dissolution modeling approach available. The first one is the direct input. The direct input is mainly used when the dissolution profile is independent of dissolution conditions. For example, immediate release product with highly soluble drug substance or an osmotic product. This method is not suitable for virtual bioequivalence trial though. However, this method is very simple as it doesn't need any additional fitting. In the second approach, which is the Weibull model, wherein the dissolution data is fitted into an empirical function first, and then the parameter derived from the equations are used for the predictions purpose. So this method is usually used for extended release dosage form where we assume 1:1 correlations. This method can also be used to generate the virtual dissolution profile. The next one is Johnson model. The Johnson model are usually used to check the impact of API PSG or the impact of different polymorphic form of the API, and where the dissolution data are not available for assessment. So in short, with this approach, we will not be able to use check the impact of formulation related attributes here. The next one is the Z-factor model. The Z-factor is basically a function of a diffusion coefficient, the diffusion layer thickness, the density, and the particle size, or the particle diameter, and is usually independent of the dissolution's pH. This is one of the widely used method where we can integrate the dissolution data mechanistically, but this method is not suitable to fit the dissolution data with other biphasic release or a dissolution profile with a lag phase, especially is for the long disintegration and to cover the plateau phase, which is due to the cone formations. Hence, this method is useful for BCS2 or BCS4 immediate release product. In case of a plateau, because of the cone formations, we can fit the initial part of the dissolution data as it describes the in vivo drug release. The last approach is the PPSD, which is basically a product specific or batch specific particle size distributions. In this approach, we extract the PSD from the dissolution data, and then those PSDs are used as an input parameter using the Johnson model for predictions purpose. The first case study is basically a case study where we want to use the PBBM concept to supersede the F2 criteria. Well, this is an extended release product having two different strengths, and the drug substance is a BCS class 1. If you look at the dissolution profile, this is the dissolution data of the highest strength, which is the B batch, and the red one is basically the dissolution data of the lowest strength. So here we observed a strength dependent dissolutions, meaning that F2 between the lower and highest strength is less than 50, and hence the biowaiver of the lower strength was challenging. So the PBPM was developed and validated with the two test batches. One of the test batches is basically a pivotal biobatch data, and the other data is basically the faster release batch, which is a pilot biobatch data. And you see the dissolution profile of those batches, the faster release and the slower release in vitro produce the faster release and slower release in vivo. So that is kind of establishing the rank order correlations. And we establish a bioequivalent safe space, which is by the modeling approach, which is between the biobatch and the virtual batch, which is presented as a dotted line here, right, from the modeling approach. And any dissolution profile in between these safe space are anticipated to be bioequivalent with the pivotal biobatch. So in our case, the dissolution profile of the lower strength is basically falling in between the dissolution safe space, and we can justify that the dissolution dissimilarity in these conditions, in these QC conditions, is not clinically relevant. And the dissolution differences which we have observed is basically a difference due to the different sink conditions, because in one of the experiment we have compared the dissolution data of the lower strength using multiple unit, two tablet of the lower strength, and compared with the one tablet of the highest strength, and this was found to be comparable. So this is basically a step where we develop and validate the model using our the pivotal bio batch and the non-bio batch. And if you look at the table here, table for the input parameter, you would observe that the disposition parameter were calculated by feeding an oral solution data because the IV data was not available. The effective permeability was calculated from the apparent permeability from an in vitro CAC2 data. The intestinal transit time were altered from the default values in order to capture the Tmax and Cmax accurately, because to capture the prolonged absorptions of the modified release dosage form and justified from the literature. The Weibull model was used to integrate the dissolution data. And if you look at the table here, which is basically, these are the observed data and these are the predicted data from the model using the dissolution profile in the QC media using a Weibull dissolution modeling approach. And if you see, the predictions error for the individual formulations is less than 15%, and of course the average predictions error is less than 10%. That means the model satisfies the criteria for the validations, and now we can use this model for its intended applications. In the next stage, the dissolution data of the virtual batch, which is pretty much faster compared to the bio batch and slower than the non-bio batch, were again fitted to the Weibull functions and use those Weibull functions to predict the plasma profile. And here is a comparisons of the predicted data of the virtual batch and the bio batch. And if you look at the T-bio ratio, it is within 20%. So as per the US FDA 1997 guidance, since the predicted outcome of the Cmax and AUC between the virtual batch and the pivotal bio batch is no more than 20%, so the bioequivalence between the virtual batch and the pivotal bio batch is concluded here. Now, since the dissolution profile of our lower strength is in between the bio batch and the virtual batch, it will also be a bioequivalent. Now, the model was developed using the highest strength, and we have extrapolated to the lowest strength because the PK was linear and the dissolution differences was observed because of the differencing conditions. The next case study is an example of utilizing the SEP space to set the clinically relevant dissolution specifications. Now, this is an example of immediate release capsules with again 2 strengths. So this is basically the dissolution data of the innovator formulations, and this is the dissolution data of the pivotal bio batch, right? And the dissolution specifications was initially set at Q 80% at 45 minutes based on the pivotal bio batch, which was challenged by the health authority, and they recommend to titrate to 80% in 30 minutes. We have used the modeling and simulations approach to justify that the 80% release in 45 minutes would be still bioequivalent with the pivotal bio batch. And in order to do that, and please remember, this is a drug substance of class 3, so the rate of absorption is not dependent on the rate of the product dissolution. But in this case, we had very limited data. So we only have a pivotal bio study data of our batch. Only one test formulation was available because in most of the cases, because of it's a class 3 molecule, the pilot data is usually not available for the generic industry, and it is very difficult to manufacture batches with a non-bio batch. So the available data was a literature data with a different dosage form with different dose level, and of course our pivotal bio study data. And since the batches with different release profile, or in the sense of different B batches were not manufactured, so the rank order relationships could not be established here. In order to create the virtual dissolution profile, it was basically done using a manually by applying different scaling factor to the pivotal bio batch, and the dissolution profile of virtual batches were created so that the virtual 1, 2, 3, and 4 releases 80%. in 30 minutes, 45 minutes, 60 minutes, and 75 minutes respectively. And with the help of modeling and simulations approach, we justified that even if the drug releases 80% releases in 45 minutes, it is still bioequivalent with our biobatch. So in this stage, the literature data of different dosage form and different dose level were used as an observed data for validation purpose. So in this stage, the dissolution data were not incorporated. Only the other physicochemical properties, let's say the solubility, effective permeability, etc., and the dispositional parameter from the IV infusions were used to create these models, right? And the effective permeability was not available from the literature, from the in vitro data, and hence we had to rely on oral solutions. So we basically fit the oral solutions to calculate the effective permeability. And if you look at the predictions error, all the formulations, individual formulations, predictions error is less than 15%. It's basically actually less than 10%. So it meets the criteria. In the next stage, the in-house batch data, the pivotal biostudy data, were used to validate the model to increase the confidence. So in this stage, we basically incorporated the resolution data in the QC media, and since this is basically a class 3 where we need not incorporate dissolution data mechanistically, the data was used as such, and the dosage form was switched to the controlled release dispersed. This is the observed data of our pivotal biobatch, wherein the test formulation was found to be bioequivalent with the reference formulations, and this is the predicted data. So that means our dissolution method is able to conclude the same B judgment considering the intra-subject variability and the confidence interval limit. And of course, the predictions error was less than 10%. In the next stage, the dissolution profile of those different the different virtual batches were integrated into the model and predicted the plasma profile. And if you see, this is the table where the T/R ratio between here the test formulation is basically the virtual batches with different release, 30, 45 minutes, 60 minutes, and 75 minutes, and the reference formulation is the pivotal B batch, and the T/R was estimated. And if you see, as per the US FDA guidance 1997, all the virtual batches were able to meet the criteria as the difference between the minimum and maximum release is less than 20%. However, if we consider the realistic scenario of our pivotal data, considering the intra-subject variability and the lower and upper bound confidence interval, this virtual 3 and 4 is going to be towards the edge of failure study, right? So in totality of evidence, using the totality of evidence approach, we concluded that virtual release profile 2 will be able to meet the BE criteria. So the clinically relevant dissolution specifications with 80% 45 minutes would still be a bioequivalent with the pivotal biobatch, and the clinically relevant dissolution specifications was justified using the modeling and simulations approach. These are the summary of my presentations. In vitro data can be integrated by mechanistic or non-mechanistic way. The best practice to use the effective permeability by fitting an oral solutions. And even if the apparent permeability is available from the in vitro data, it is always recommended to fit it using an oral solution profile if available, because there is always a lab-to-lab variability was observed. In a case we want to use the parameter sensitivity analysis for the physiological parameter, especially for the modified release product, it has to be performed very carefully and scientifically justified. The BE safe space can be extended beyond the knowledge space for the low biopharmaceutics risk product, because for the low biopharmaceutics risk product it is very difficult to manufacture batches with the non-BE. And in most of the time for this kind of product, from the generic perspective, we don't usually do a pilot biostudy data. So the release in vivo release data with multiple formulations are usually not available. The BE safe space are very useful for product lifecycle management, and it is usually decided based on the minimum maximum predicted ratio less than 20% or a virtual B trial along with a parameter sensitivity analysis. These are the references which were used during the presentations. Thank you for your kind attention and wish you a great rest of the workshop. I'll be joining the panel discussion shortly.